Studying coevolution of hydrological and biogeochemical processes in the subsurface of natural landscapes can enhance the understanding of coupled Earth system processes. The overall goal of this method is to excavate a soil lysimeter to understand subsurface hydrological, geochemical, and microbiological heterogeneity.
This method can help answer key questions in the field of coupled Earth system processes, such as how do hydrological and biogeochemical processes co-evolve in the subsurface of natural ecosystems.
The main advantage of this technique is that it can capture the spatial heterogeneity of hydrological, geochemical, and microbiological properties of soil in a submeter scale.
Divide the lysimeter into voxels of fixed length, width, and depth using a Euclidean space coordinate system. Divide the total distance along each direction into an adequate number of equally spaced intervals. Assign each sample a unique XYZ location and identify it as a voxel. In this excavation, X denotes the location along the width of the slope, Y denotes the location along the length of the slope, and Z denotes the location along the depth of the slope. The size of the intervals within each dimension determines the width, length, and depth of the voxels. Shown here is the division of the lysimeter after determining spacing intervals, along with the chosen origin for the X, Y, Z system.